Okay, thank you all for the introduction. And I'm very happy to have this chance to share with you the work I've been doing these days. And before I really start, so I think some of you might hear of me talking about this topic in Hangzhou, but because we are on the revision and we have a lot of updates, so many new feedback, new takeaways for you. So don't worry about that. And before I really start, I would like to introduce a bit about where I'm currently working at. So I'm in Guangzhou National Lab here. So we are on the island in Guangzhou City, and the island looks like this, and this is how our institute look like. It's surrounded by the water and have a lot of trees with really good scenery. And I will start my talk today with a really hot topic, like the foundation models. So based on the success of the transformer, the structure applied to the AI field, and we find that using this type of model structure, we are able to somehow learn a really comprehensive way, have a really comprehensive way to learn information in a text or the image, and it helps us a lot with a lot of new tasks. And researchers, biologists, like they think maybe we can apply the structure to the biology sequencing data as well to see if we can take advantage of it and have a foundation model. And based on that, we can do a lot of other downstream tasks. And we do find a lot of new exciting results that when using this structure, we kind of can have can learn a pretty good model that we find it can do a lot of tasks like cell annotation and communication like that. Among all these downstream tasks, one of it just stands out is that the perturbation effect modeling. And this task is pretty important because not like the cell type annotation, like if you have a standard pipeline, um, you can have the information of the cell with no like this deep learning structure. And for the cell to cell communication, you can have the information of that with some traditional method as well. But for the perturbation effect modeling, usually if you do not have a big model that have a lot of information learned with intrinsic biological information within a cell, you might not have a good effect, good performance. So we are quite interested into this task. And in recent years, there's a lot of other researchers, they also find this perturbation modeling is a really important application. And they all think that this is a really important milestone if we want to build a virtual cell. Like if you and this is a really important task for us to test if the model really learns the truth in a cell. For example, here, if you can have, for example, you just in silico perturb some of the genes and you can predict the consequences correctly, which means that it will help you to save a lot of time and money in doing experiments. And based on that, after we have the virtual cells, we will be able to build virtual tissue and virtual human beings. But let's look at how what exactly is the perturbation modeling process is. So usually this process can be defined as that you have first you have a population of cells, and then you treat them with, like, for example, you treat them with different CRISPR screening, or you use some different types of drugs to treat them. And after that, you have the treated population of the cells. And you just use a lot of data available and to train a model. And based on this, you will be able to, for example, you have a new perturbation, you will be able to predict what will happen, to predict the after state. And our first goal was try to have a look at how those foundational model performs. And another goal we were thinking about is that besides that, we want to see what's the other tools available for us, how they perform, and does the foundation tools really outperform them? And we had a look into the research and to see what methods are available for us. And after taking a look at that, we found this field is quite complicated, and the available methods, they have different tasks that they want to take care of. Like we divide the tasks they take care of into three. And here you kind of see that the first task is that some of the method, they would like to make the model having the ability to predict the effect of some unseen perturbations, which means that in a training data, some of the perturbations are unseen, and uh, and you want to predict those strings that are not available in the training data. And the second scenario is that some of the methods, they would like to take care of the perturbation interaction effect. So which means that in the modeling, in the training process, they might have a large data, and most of the perturbations are single perturbation. But what they want to predict is a combination of the single perturbations. They want to know the interaction of the genes. And this third task they had a look at is a cell state transfer, and this is a most popular task that a lot of methods have a look at. So this task, you really have, for example, because it's easier to do the perturbation experiment on the cell line cells. So you can train a model with a lot of perturbation data on the cell line cells, but transfer that to other cell types like the PBMCs or fibroblasts like that to see what will happen on those different cell type cells. So they generally they look at different tasks, and there's no method that available for us to do all of these tasks in just one method. But after having this knowledge, we were thinking maybe it's good for us to do a benchmark study to see on for each task how they perform, and is there any space for them to be developed, to be combined into a single one method that can deal with all these three tasks. So because we want to do a benchmark, and we had a look at what's available in the research papers, and we found most of the benchmarks are usually provided with a method paper, and they sometimes for most of the time they only used like 3 or 4 data sets, or sometimes even just one, and also they only compare their method with some very popular one. They do not compare their method to the current or available method. So we think it's a good time for us to do a really comprehensive benchmark study. So we try to collect all the available perturbation data sets, like nearly all the data sets, single cell data sets with CRISPR screening. And we also included some big data that are released recently this year with a lot of drug used on the different cancer cell line cells. And this is how the public data we collected look like. And besides that, we also generated 2 in-house data, because we found that in single cell data available for us, most of the cells are PBMC, and we think it might be better if we can have the perturbation treated on the PBMC as well to see how it looks. So we have 2 in-house data, and the first one we have the cell line cells and PBMC organoid treated with drugs, and another we have the PBMC treated with more a larger amount of drugs, and all of them we use the treated and untreated cells together to do the RNA sequencing, and this is how our data look like. And after collecting the data, we were thinking what evaluation metrics that might be a good start for us to have a look at how those model performs. So we think about 4 aspects, and we divide, and here it's just a plot to show you which aspect we have. So the first one, we want to have a absolute accuracy. And here we use some R square and MNC method metrics like that. And another, so this is more for us to check the reconstruction of the model. And the second is to have a look at the relative effect to see if they capture them well. And we have the Pearson correlation of the delta, and also we have the direction agreement accuracy calculated. And to make the task even more strict, we also want to have a look at, so after having a relative effect captured, we want to have a look at if the method could, the generated data by the method can be used to compare to the control data to see if the differential expression could be recovered, and we calculate the accuracy and also the effect size to see if there are any issue with that. And besides that, the fourth metric we are quite interested to see is to see the distribution of the data generated, to have a check for this is also for the check of the reconstruction quality. So as I just mentioned, there will be three tasks that the methods are taking care of. And the first one of them is to predict the unseen perturbations. And the process would be that in a training process, they would have a lot of perturbations, but for the one they're interested in are not included in the training data. So they want to know what will happen to the unseen perturbation. And here we just use the metrics that I mentioned to evaluate the available methods. And we also have no perturb here, means that we just collect the control data to be compared with the others. And the baseline here is a simple MLP model generated by us, and the other colors here represent different methods. And the SCGBT and SA foundations are fine-tuned foundation models to do the perturbation task. And as you can see here, if we look at the top 22 genes Pearson correlation using their overall expression, we find that all of the methods, it performs pretty well, even the baseline. And most of the scores they have are above 0.95, which is pretty good. And we could not tell a very big difference between those methods. But if we look into the detail of how these methods perform and across different data records, and this is a result for within one of the perturbation data, and we calculated the effect size for different perturbation. And here, ranking the data by ranking the record here by their perturbation effect size. And here, this one have the largest effect, and this one have the smallest effect. And we find that although we could not see a very clear difference based on the Pearson correlation score here, but if we look into the detail of it, we find that with a higher effect size, the evaluation score we have will be higher, which means that the bigger effect is easier for us to be captured. And this is still at the stage that we are just having a very not that strict evaluation. And after we use the metric 3, which is using a more strict way to evaluate those methods, that we want to calculate do the differential expression analysis between a generated predicted data with the control data to see if they can help us to retain the DE genes. And this is because if you want to do the differential expression analysis, it really like requires a method to have both of the average expression to be recovered, and also the variance modeling should be at a good state as well. So we can see that if we do the differential expression analysis, most of the methods, they do not perform that well. except for the perturb net. This one can achieve the 0.56 top 100 accuracy for the DE genes, but for others, nearly all of them, they would return, like for example, you have 5,000 genes for the whole data, but they would have variance modeling really bad, so they return like 4,000 genes, like 80% of the genes to be DE genes. And the rank of the, because we're only calculating the accuracy for the top 100 DE genes, so because they just return nearly all of the genes to be DE, and the top 100 rank would be really bad. So the performance here we see here are really not that good. And we are, besides that, we find that if the data have larger effect size, usually the method will provide you a better result. So this is aligned with what we see before. Like if the effect size is larger, it might be easier for the methods to have a good result. So this is for the scenario one that we were trying to look at the perturbation response prediction on the unseen perturbations. And next, we move to the scenario two, that we want to have a look at how this model performs when trying to predict the interaction of the perturbations. So here, this task generally looks like this. So in a training, you have most of the training data to be single perturbation, and you want to predict a combination of those perturbations. And this task can be divided into 3 sub-scenarios. And here, for some of the training process, you might have not seen any of the single gene before, but for some of them, you might see one of the perturbation before. And for some of them, you see both of the single perturbation, and you want to know the combination of them, like what's their effect. And this makes this scenario quite complicated, because if you are trying to evaluate on a scene 2 scenario, which means that this model only needs to have the ability to know the interaction of the genes, and they could use that knowledge to predict a combinationary effect that they would provide you a good result. But if it's a scene 0 scenario, which means that the model has to do 2 things. The first one is to have the generalizability to predict the unseen perturbation first, and after that, they need to have the ability to know how those genes interact with each other, and try to give you a like what will happen if they are both perturbed. So the scene 2 scenario is a much harder, more difficult task compared to the scene 2 scenario. And besides that, this task, when we're looking at this task, we need to take care of the additive type as well. So here, we would divide based on the data we have, we divide the additive type into 2, the buffering and synergy, which means that based on the true data we have, we would look at how the effect look like compared to the combination of the effect that we have on the A and B together. If it's smaller, if it has smaller effect, it will be defined as a buffering effect. And if it's larger than the combination, it will be defined as a synergy. And after that, we had a look at how those methods performs and what the results are given to us. So here, we have the scene 0 and scene 2 compared in these 2 fixed boxes, and we also color the boxes by the buffering and synergy. So the first takeaway is that when we just look at the buffering and synergy, we find that the buffering effects are easier for the methods to capture them. And when we look at why the synergy additive scenario does not have that good performance, we found that usually the model would generate a really conservative result, that they do not like to predict the effect to be larger to predict the effect always smaller than the combination of the perturbation that we think. And this is for the additive type. And if we compare between the scene 0 and scene 2, these two scenarios, we find that for the buffering this type, the scene 0 and scene 2 does not have that big difference. But for the synergy, the scene 2, somehow they have more information available, so they might perform a little bit better. And this is a result we have based on the perturbation interaction prediction, the task 2. And when we move to the task 3, because this task, it does not really need model to have that good knowledge to modeling the gene interactions. They really just need to have a look at how the perturbation how the perturbation embedding looks like, and they just need to know the interaction between the cell type and the perturbation. So the methods here we use in task 3 are completely different from the first two tasks. And for this scenario, we had a look at how those method performs, and the conclusion we have are quite different from the first two. Is that when we look at the data, so here we calculated so the E distance here means that the cell type distance within the data set. So here the different columns represent different data sets, and we find that across different data sets, effect size are pretty different, and we rank them, and we found if the distance between the cell types are larger, it would provide you not that good result. But if the distance between the cell types are smaller, if you the cell data transfer models provide you a better result. So so based on the scenario 3, the conclusion is that this is more the performance of the methods are more dependent on the cell type distance. So this is not that a pretty different conclusion from the first two tasks. So after talking about all of the result, the takeaway message we have now is that like a good mean does not mean a good biological signal. So for example, on the first scenario, we found that using a Pearson correlation and the cosine similarity like that, we found most of the methods, they perform pretty well. Like if you could still remember that most of the methods, they can provide you a performance of Pearson correlation with a score like around 0.95. But if we look at the DE genes detection, except for the PerturbNet, all of the methods, they perform pretty bad. And just based on that, we found that the variance must be modeled pretty good. Like because only the PerturbNet, that method, it takes care of the distribution of the data to take care of the variance. But for the other method, like the VAE based or the fine-tuned foundation models, they do not deal with the variance well, so they could not return us the true DE genes. And other takeaways from the results here is that in a combinatorial perturbations, we found that synergy effects are harder to compare to the buffering effect to be recovered. And also we found that on the scenario 3, the cross-cell type transfer is somehow bottlenecked by the domain shift. Like we still find that when the distance between the cell type is larger, it's more difficult for the data to provide you good results. So which means that we still need to focus more on how to adapt those strategies, but not just calculate a perturbation embedding and just add it to the embedding of the cells like that. The methods still have a lot of space to be improved. And that's I just talked about the results and some findings we have about the perturbation benchmark we have, and based on that, we generate a website which includes all of the information we have about the data and all and the evaluation results we have, and this is available here, and it provides interactive ways for you to have a look at how those method performs, and we also provide a pipeline that if you have new method, you want to compare it with evaluation we did, you can generate a new comprehensive result as well. And besides that, because we found in this benchmark that the discovering of the DE genes using a predicted perturbation response data is really difficult, and we were thinking maybe we could try to build a agent system to do this using an LM, and we are trying to just focus on the recovering of the DE genes and see if the agent system can help us to do that. And this is another work we are currently working at. And next, I want to thank our lab head, Louis, and other people that put a lot of effort in this work, Lanxiang and Yunling, and other collaborators that I have. And thank you. Thanks for listening.